Welcome to the first endocrine lecture. In this lecture we're going to learn about how cells communicate with each other, and we'll learn a little bit about the chemistry of hormones. In this figure you see the four different ways that cells communicate with each other. Direct communication through gap junctions, paracrine communication where cells secrete chemicals that reach other cells, endocrine communication which occurs when hormones are secreted into the blood, and synaptic communication when neurotransmitters are secreted directly on the membrane of their target cells. Indirect communication, the ions and molecules move from one cell to a neighboring cell through gap junctions. There are very few examples, but they are important. We'll study this when we get to the heart. In paracrine communication, cells secrete chemicals that activate cells nearby. The chemicals simply diffuse through the interstitial fluid. This is important in immune activation. Endocrine communication allows chemicals to be broadcast throughout the whole body. So in endocrine communication, chemicals called hormones are secreted into the blood and are transmitted everywhere throughout the body. Synaptic communication is a specialized communication of the nervous system. Chemicals are brought directly to the target cell and are secreted there. A target cell is a cell that has a receptor for a particular chemical messenger. In the endocrine system, target cells may have receptors on their membranes or on the inside of the cell. The job of a hormone is to regulate the mechanisms inside of a cell in some way. They may cause the cell to produce new proteins, they may change the metabolism of the cell, or they may activate membrane channels and allow ions to flow. In general, the endocrine system regulates long-term or steady-state processes. In contrast to the nervous system, which causes changes in the course of milliseconds to seconds, endocrine changes take minutes to hours. The shortest endocrine response may just be a few minutes, such as the secretion of epinephrine with your fight-or-flight syndrome. Okay, now we're going to talk about the chemistry of hormones, and this gets a little involved, and I'm going to try to keep it as simple as possible. We do divide hormones into three chemical groups: amino acid derivatives, peptide hormones, and lipid derivatives. We're not going to focus too much on the chemistry of all of them, but we do need to look at some particular aspects of how the chemistry affects the secretion and the activity of the hormone. First, let's look at the amino acid derivatives. I'm not going to test you on which amino acid they're derived from, but I think it might be helpful for you to have that as a reference. The amino acids tyrosine and tryptophan can be converted into hormones. The hormones that are derived from amino acids you do need to know. You just don't need to know which amino acid they're derived from. So we have the thyroid hormones, mostly referred to as thyroxine, T3 and T4. We have the group called catecholamines. These are also called biogenic amines, and they include epinephrine and norepinephrine. And then finally we have dopamine, serotonin, and melatonin. The peptide hormones are made like any other peptide. You put a bunch of amino acids together into a peptide chain. Some are short and some are long. Some have a little carbohydrate group attached, making them a glycoprotein. This changes the duration of the lifespan of the hormone, and it may change the way that it's carried in the blood, or it may change the way it is metabolized in the body, but that's beyond the scope of the course. So we're going to learn all of the peptide hormones together and not try to make a distinction about which ones are short or which ones are long or which ones are glycoproteins. It does help you to know that many of them are secreted as prohormones. Prohormone is a precursor to a hormone. If it is converted to the active form before it's secreted, then we don't really care. If it's secreted as a prohormone, though, then you may need to become aware of how the activation occurs. I can think of a specific example of that, but we'll save it until we get to where we need it. Lipid derivatives are usually divided into two categories. The eicosanoids are actually paracrine factors, and they're included here just to make sure that we have a very thorough discussion of all of the cell-to-cell communication types. But I'm not testing you on eicosanoids because they're not hormones. The steroid hormones are the only lipid-derived class that you need to know. And you do need to know which ones are steroid hormones, but that's a very simple list. There are only 3 sex hormones that you need to know, which are steroids, testosterone, estrogens, and progestins. The adrenal cortex secretes steroid hormones. All of the hormones from the adrenal cortex are steroids. So if you know which ones those are, you know they're steroid hormones. And last but not least, the kidneys secrete a steroid hormone called calcitriol. Calcitriol is actually derived from vitamin D. What difference does it make whether they're lipid hormones or not? Well, it makes a difference in how well they're carried in the blood. Hormones that are lipid soluble, that is, they're hydrophobic, they don't mix with water very well, tend not to be dissolved in the plasma very well, and so you need to have an alternate way to carry them through the blood. So we say that hormones are either free or bound. They're free if they are dissolved in the plasma. Free hormones tend not to stay dissolved in the plasma very long. They're going to diffuse out, they're going to be removed by liver or kidney or broken down. On the other hand, bound hormones, which are the ones that don't mix with water very well, tend to circulate in the blood for a longer period of time. Now, thyroid hormones, even though they are amino acid derived, happen to be hydrophobic. That is, they dissolve better in lipid than they do in water. And that's an important thing for you to remember. We'll look at why that is in just a minute. But for now, we need to know it's the thyroids and the steroids that tend to be bound. Most of these hormone particles are bound to plasma proteins as they circulate through the blood, and that makes them last in the bloodstream a lot longer than the other hormones. Okay, let's get our hormones circulating in the blood, either free or bound, to our target cell. A target cell is a cell that has a receptor for that hormone. Remember that the hormones are in the blood, they're distributed throughout the whole body, so the only way to make sure that the cells that you need to respond to the hormones are able to respond and the other cells are not, is that the target cells are the only cells that have the receptor for the hormone. This receptor can be either stuck in the plasma membrane or it can be in the cytoplasm or nucleus of the cell. If the hormone is water soluble, that's our proteins and our catecholamines, then that hormone has to have a membrane-bound receptor. It can't enter the cell if it's water soluble. On the other hand, our lipid-soluble hormones, remember which ones those were? Thyroid and steroid hormones. Because they're small particles that are lipid-soluble, can diffuse right through the phospholipid bilayer, and those are the ones that have intracellular receptors. So, catecholamines and peptides have a membrane-bound receptor. They actually attach to the outside of the cell and they never enter the cell. We have then an intermediary on the inside of the cell for these hormones that do not themselves come into the cell. We say the hormone is the first messenger and that in the cell somewhere there is a second messenger, something that's activated by the hormone binding on the outside. So here in this diagram you can see that this is the plasma membrane here. Here is the hormone coming in from the blood. It binds to its receptor in the membrane, and when the hormone binds to the receptor, it's going to activate some process on the inside of the cell, and as you look down here, you're looking inside the cell, you can see that whatever it activates, it's going to cause changes inside the cell. The hormone never enters the cell. The second messenger is going to cause the changes inside of the cell. Some examples of important second messengers that we've identified include cyclic AMP, cyclic GMP. AMP and GMP are both derived from nucleotides, adenosine and guanine. And a third one is calcium ions. Now, these processes are pretty complicated, and there's a whole network of second messenger systems within the cell. So these are just examples and they're not even a complete story. We're just getting the barest of introductions to the whole idea of second messengers. And one of the hallmarks of a second messenger system is that the message is amplified inside the cell. When that hormone binds to the outside, it can continue to activate the second messenger molecules until thousands of those active molecules are created inside the cell, causing a widespread and rapid change within the cell. Furthermore, the effect is immediate. So when a hormone binds to a membrane-bound receptor, it instantaneously transforms the metabolism of the target cell. But also, as soon as the hormone leaves, which it will, getting broken down by an enzyme usually, then the effect is instantly turned off. Now, membrane-bound receptors are subject to a pretty common physiological process called up and down regulation. In down regulation, we reduce the number of hormone receptors. If a hormone is secreted excessively, then you tend to down-regulate the number of receptors to kind of turn down the response. And obviously the opposite is going to occur with upregulation. With upregulation, if you don't have a lot of hormone available, you increase the number of receptors so that even a small amount of hormone can have a large effect. Now, a word about G proteins. We're not going to get bogged down into all of the details of G proteins in the book, but I think it's important for you to recognize the term. When a hormone or a neurotransmitter binds to its receptor, it may activate a G protein complex, which then can activate cyclic AMP. And that's going to be the second messenger system. G proteins can also, in other cells, stimulated by other hormones, activate calcium ions. Those calcium ions are either coming in through the outside of the cell, through open channels, or they're being released from intracellular stores. Okay, for our lipid-soluble hormones, the ones that have intracellular receptors, the receptor inside of the cell binds to the hormone and goes to the DNA in the nucleus. It specifically triggers protein synthesis. So here you can see the effect of a steroid hormone. Notice that it goes right through the plasma membrane. It binds to its receptor. Sometimes that receptor's in the cytoplasm, sometimes it's already in the nucleus. Either way, it has to get into the nucleus to do its thing. It's going to bind to the DNA. It's going to activate a gene. It's going to increase the synthesis of a particular protein, and that protein synthesis then alters the activity of the cell. Now, in contrast to our membrane-bound receptors, remember what they did? They activated a second messenger immediately. This kind of hormone has to get into the cell, turn on protein synthesis. It's going to be at least an hour before the cell shows any effect at all. So that's the steroid hormones, and we see a very similar situation with the thyroid hormones. The thyroid hormone enters the cell, it binds to its receptor, the receptor can bind to the DNA and cause protein synthesis to occur. However, thyroid hormone has another effect. Interestingly, thyroid hormone binds to receptors on mitochondria, and that process then increases not just ATP production, but increases the utilization of ATP. ATP is used up, and in the process produces a lot of heat, and that heat helps maintain your normal body temperature. Now, how are hormones controlled? We know that they're produced by cells in the endocrine glands, but you have to have some way of imagining that the cells know when to produce their hormone or when to secrete their hormone. And so we have what we call endocrine reflexes. And these are very similar to neural reflexes. You have to have some stimulus and some response. You have to have a control center that is determining what the stimulus is and how to respond to it. So in the case of endocrine reflexes, generally there's going to be some trigger that causes the secretion of a hormone, and that's our reflex. Some of the hormones are actually secreted by nerve cells, and we call these neuroendocrine reflexes. We'll see some of these as we go through the rest of this chapter. And here are the different categories of triggers that can cause the secretion of hormones. A humoral stimulus is a chemical in the bloodstream. Some examples of that. When you eat sugar, you secrete more insulin, so that sugar is a humoral stimulus. When you eat calcium, you secrete calcitonin, so calcium is a humoral stimulus. Even water and salt can be humoral stimuli for the hormones associated with their regulation. A hormonal stimulus occurs when one hormone stimulates the secretion of a second hormone. So we see that, for example, with thyroid stimulating hormone, which, as you can imagine, circulates to the thyroid and stimulates the secretion of the thyroid hormones. The third category is neural stimuli. If the nervous system triggers an endocrine response, that would be a neuroendocrine reflex. That neural stimulus caused the secretion of the hormone. An example of that would be the secretion of epinephrine when you're stressed. Now, I didn't see this in the book, but I think it's very useful information. There are 3 patterns of secretion. Some hormones are secreted basically all the time. They're chronically secreted, just sort of in a steady state pattern all the time. Examples of this include thyroid hormone, which you need to maintain your normal metabolism, growth hormone, which you need to maintain protein synthesis, and in males, testosterone secretion is steady state. A second category is acute. You secrete hormones acutely only when an abrupt stimulus from the external environment caused the need for that hormone. And an example of that, again, is epinephrine secretion with your fight or flight, melatonin secretion, which it's controlled by light and dark cycles. Insulin is secreted when you eat, particularly if it's high in sugar and amino acids. Aldosterone is secreted when you have a high level of potassium. Our third category is cyclic. If a hormone is secreted in just very large swings, very high secretion followed by low or no secretion, that would be a cyclic pattern. And the classic example of that is the female sex hormones. This concludes our overview of the hormones. The next in our series of endocrine lectures will go through the different endocrine glands one by one, talk about the secretion of the hormones, the effects of the hormones, and the regulation of the hormones. See you soon.